presentation of the speaker, Charlie and Pierre. So we have 2 sessions on spike sorting. So I'm supposed to introduce the 2 speakers. So I'm excited because I spent many time to try to make a spike sorter, and I did not succeed. So if you play with extracellular signals, you know that the tools is dominated by KiloSort. Anyone is using KiloSort. But KiloSort now have 2 main competitors, and we are lucky because we have the 2 main competitors in the room, which is maybe Charlie and Pierre. They are 2 main, and they work a lot to understand the detail of algorithm, and they will present each one a solution maybe that could replace one day KiloSort. So we are super proud to welcome Charlie first, which is in the Paninsky lab in New York, and then Pierre, which is from Lille, France, in an INSERM lab. And so, well, no more to say. That's it. Merci.
Yeah.
Come on, time.
It's my first time with one of these microphones, but is it working? Okay, great. Yeah, I guess I like Sam's adversarial phrasing as a joke, but to me it's a collaborative field. So but thanks for the intro, Sam, and thank you guys for being here, and thanks to the organizers of this conference. Yes, I'm really excited to be talking at this conference just because, first off, like as a PhD student working on tools and methods in electrophysiology, I think it's amazing that there's a community like the one that's kind of growing around spike interface. It's a great thing for me because I know that if I come up with a method or something like that, there's a place where I can put it that makes it easy for people to use it without tearing their hair out, which is something I'm still trying to do. So it's a work in progress of getting all of our tools in there, and I'm slow at that. So I'm sorry to these guys, but that's the goal. And I guess that's why we're here. So it's a pretty exciting thing. And that's part of, a bigger culture of open software and data that I think is amazing in this field. Shouts out to the International Brain Lab. They have a really cool, easy to use data infrastructure, which makes it possible to test out methods and algorithms at scale. And also it's really important to develop methods and algorithms that can handle data at the scale that they're working with. And also I want to thank some amazing collaborators. I'm going to be showing data today from some presenters from this morning who talked about it. My presentation will be a bit of a joke on that data compared to theirs. But I'm coming from a different perspective, I guess, of developing tools. But yeah, so thanks to those collaborators and some more that I'll talk about pretty soon. So yeah, in this field, we have these large scale multi-region recordings. This is an example from the IBL. They have tons of recordings across the entire mouse brain. We have local field potentials. We have action potential recordings. I'll show some data later from monkey and even from humans. So in this field, I think it's really important to develop methods that are kind of robust to all of these different conditions, different kinds of probes. There's ultra high density probes, NeuroPixels 1 and 2, and all of these other silicon probes. So we want to kind of come up with methods that can deal with the scale and kind of variability of all of these different kinds of data. And I think that there are a few challenges in that, and there are some specific ones that I'll kind of highlight today, but obviously there are way more challenges than just these. So some of the things we're thinking about in our group, first and foremost, is probe motion artifacts, which can happen when you're doing these recordings. They're not present in all, high density multi-electrode array recordings by any means, but they're there in some cases. And we want to come up with robust ways to deal with that. Another issue is, at the single unit perspective, even just beyond drift, there can be activity changes, waveform changes, and amplitude changes over time in single acute sessions or over days in a chronic setting. So that's something that's hard for these clustering algorithms to handle. And then also, I think right now there are some kinds of brain activity that are maybe somewhat out of reach for these tools that we have right now. Maybe I'm wrong about this, and I would love to hear about it if I am, but I think that there are some brain regions, like I'm thinking of cerebellum, which are just really challenging for spike sorters as they are right now. I don't have the answer to that at all right now, but I think that's an important open question. Maybe Olivier will talk a little bit about that, I'm sure. Okay, so first case, the main thing I'll talk about today is probe motion. So even in acute head-fixed mouse recordings, there are challenging cases with probe drift, but I'm also interested in kind of a broader collection of recordings. So we could think about the problem of tracking the probe's motion across days in a chronic implantation. Usually those are super stable, but there can be some kind of settling of the probe, especially at the tip of the probe, that causes issues. Here I'm showing some data from a human recording from the Chang lab at UCSF. And I'm going to be showing a lot of plots that look like this. So I just want to talk through what this is. So this is something like a spike raster like you see in spike sorting, but it's an unsorted representation of the spiking activity. So on the x-axis, we have time, and the y-axis is the depth along the probe. And each pixel here is sort of a representation of the firing intensity in that spatiotemporal bin. So to make this plot, we kind of detect a lot of spikes and localize their positions in space, which I'll talk about more. And that helps us make these kind of spike rasters. So here, in this particular one, you can sort of see these three horizontal lines, which are not what I want to draw your attention to. I think these are probably just dead channels or something that I didn't correctly remove. What I'm interested in is here, this big blob of activity. It looks super diffuse. It's really hard to say what's going on there. Is it even probe motion or is it just noise? But actually we'll kind of show that there are several really clean and possible to easily isolate units hiding in this cloud once you correct for the motion. So that's what I'll talk about for the next little while. So then the other problems I was thinking about, more on the sorting side than the motion correction side, is how can we kind of deal with spike sorting when there are non-stationarities or changes over time in the unit waveforms. So this is an example, I think this is an old example from, I think, a kilosort sorting of an IBL recording, where a unit, it has an amplitude profile which changes quite drastically over the course of the recording, and that's causing the cluster to be split. In the bottom right here, I'm showing a visualization of some spikes detected in a NeuroPixels Ultra recording. So they have a collection of recordings that they made, which featured a lot of drift because they had actually imposed the drift on the recording to study how those probes can handle these motion artifacts. So this is not a typical NeuroPixels Ultra recording. So in the bottom right here, I'm showing the same graphic from the last page, where the x-axis is time and the y-axis is the depth along the probe. And I'm also plotting an estimate of the motion over time overlaid on top of that spike raster. And then these other two panels, I might show these again later in some different contexts. This is sort of the horizontal and vertical positions of the spikes, and they're colored by the spike amplitude. And we've got the probe kind of overlaid on top of these horizontal and vertical positions, just so you can see where the spikes are in space relative to the recording contacts. And here we also have an amplitude profile on the x-axis. So what you can kind of see here, maybe it's hard to see, but there are some kind of cleanly identifiable unit-looking things that you can see in these plots. And they have amplitude profiles that are changing really drastically. They're moving across the entire probe, and this can play havoc with typical clustering algorithms. So it's sort of an edge case from spike sorting, but you could imagine that if we had an algorithm which could handle these kind of problems, that's going to have to be a robust algorithm that should hopefully work in a lot of cases. And then just the last case, I probably won't show these right now, But IBL has this really cool visualization website, which maybe Olivier will show, where you can dive into all of their recordings. You can see the raw data coming off of the probe. You can see the spike sorted units. It's really cool, and I recommend people check it out. This is an example from olfactory bulb in mouse, and you can see that there's this super dense firing activity. And these are the sorted units that come out. And basically, you end up with something like 500 units detected here, and then only 10% or so pass the IBL quality control metrics, which is basically what you would expect to pass just by chance because of their p-value threshold. So essentially, the sorting in this region is really hard to do, and this activity is just so dense. I think it's a big challenge. Okay, so our goals are, number one, to build robust and easy use tools for these different recordings. And to me, easy to use means they integrate with Spike interface. And hopefully over these few days, everybody will agree that that's what easy to use means. So the tools I'll talk about is one, DREDge, which is our motion tracking algorithm, and also DARTsort, which is a work in progress drift aware spike sorter. It uses kind of an alternative approach to drift correction in the spike sorting. And it's also kind of like a GPU enabled software toolkit that's built around spike interface. Okay, so probe motion. I'll just start by kind of talking about the steps in our algorithm and how it works. So the first step in any motion estimation algorithm that operates on spike data is first you need to detect some spikes. Once you have the spikes, we apply some denoising operations to clean them up a little bit, and then we localize the sources of those spikes in space relative to the probe. There are several different ways of doing that. Our favorite method is this method point source localization, which sort of models the spike as if it came from a point source in space, which is a extremely crude approximation, but it can pick out sort of a meaningful physical position for individual spikes that in the aggregate is helpful if you want to track the drift of units over time. So here's an example of the kind of denoised spike waveform that we would input into our localization model. And the way the model works is something like this. We detect this spike on several channels, and we observe amplitudes. You could measure that in different ways, maybe peak-to-peak amplitude, or just the absolute value of the trough of the waveform. And here I'm kind of representing those amplitudes as vertical lines in over this 2D representation of the probe. So here, the two spatial dimensions here are the probe dimensions, and I'm showing the contacts as gray squares, and the vertical dimension is amplitude. And then we have this model, and the model says there's a point source out there, it has a certain brightness, and it exists at a certain spatial position X, which has certain distances to all of these different contacts. And based on the model, we predict that there should be some amplitude that we observe on all of the contacts of the probe. And I'm sort of representing that model distribution of amplitudes with this surface here. And we try to fit that surface to the spike data. And we do this for every single spike to determine spatial positions for all of the sources that emitted them. Okay, so once we get that, we can then compute one of these spike rasters that I was talking about before. So here, again, the x-axis is time and the y-axis is depth. This is coming from an IBL recording. I would say that most IBL recordings are less drifty than this one. So this is like an interesting example where there's, some parts of the recording are quite stable, and maybe other areas have this drift happening. Down here, there's some roughness in the activity pattern. So this is kind of an interesting example of non-rigid or spatially varying probe motion over time, or brain tissue motion relative to the probe, I should say. So the current main method for estimating motion is what's implemented in DREDge. So just to quickly go over what they do, essentially the first thing they do is they make a template. So this is sort of a representation of the activity as a function of depth. So here I'm visualizing an example template. So this is something that's over depth and it has a sort of no width in space. So one way to do this is to take the mean over time of the spike raster, and that gives you this function of depth, which they call the template. Then what happens is DREDge will cross-correlate the template with all of the time bins of the spike raster. So by cross-correlate, I mean it'll take that template and sort of slide it vertically up and down every single frame of this spike raster and kind of find the position where it best matches the raster. And that gives you an estimate of the displacement of that frame of the raster relative to kind of the global activity pattern. And once you do that, if you collect all of those different displacements for every single frame, you'll collect them into an estimate of the motion over time. So this kind of vector, which has time on the x-axis and displacement on the y-axis. And they can kind of repeat that procedure multiple times to refine the estimate. And this works pretty well most of the time. So our approach is a little bit different. And I think the idea is that DREDge's approach is a centralized one, where you try to model the entire recording with one global template. And that just doesn't exactly always work. There's not always one template that can model an entire recording. Our approach kind of comes from a different angle, which is to be decentralized. So rather than using one global model to try to capture the structure of the recording, we'll use local information. So here's how that works. Essentially what happens is we have a similar spike raster, and we look at all pairs of time bins or frames of the raster. So, or in other words, just columns of this image. So here I've picked one pair of time frames, this time bin in red and the one in blue. Down here, I'm plotting them as functions of depth. So what was on the y-axis of this spike raster, now I'm visualizing as the x-axis, and the intensity of the raster is being plotted in these red and blue lines. So this is a pair of time bins of the spike raster. Then I'll cross-correlate them in the same way that DREDge cross-correlates its template with all frames of the spike raster. And that gets me an estimate for the optimal relative displacement of these two frames. So here it happened to be 37 microns. So this is an estimate. My cross-correlation is telling me, I think that there was something like 37 microns of drift from time 3250 to time 4000 in this recording. So we'll do that for all of the pairs of time bins. And by doing that, we get a big matrix with time on the y-axis and time on the x-axis. So for every pair of times, we get an estimate of the displacement. And here it sort of looks how you would expect. So you can see that there's some upward motion visible in this portion of the recording. And that is being reflected here, where if you look at sites in this matrix, where one of the entries is larger than the other, the displacement value is positive, and on the other side, we get a negative displacement value. So it's saying there's upward motion evident here. And the cross-correlation operator also gives us a nice and useful number, which is how well the two different time bins matched across that period. So for every pair of time bins, we also get this number indicating how good of a match those time bins were. So if the activity totally changed, then we would see that showing up as a low correlation, and it might tell us, hey, don't trust that displacement value you got because you're looking at completely different neurons now, and who knows if they really are displaced by 37 microns. But here they have a relatively high correlation because the activity patterns are similar. Then the last step is we kind of go from this local information back to global. So we solve this thing called the centralization problem. And our goal is to pick one displacement value for every time bin, and we want the relative offsets of our global displacement trace to match our observed local relative displacements. So in other words, we want our displacement estimate between time t and t prime to be close to our observed estimated offsets between those time bins. So we kind of are able to extract a motion trace, which I'm plotting here twice, once horizontally and once vertically, so that I can visualize taking the relative difference at two different time bins for our global motion estimate and comparing it to this observation that we got from cross-correlating those frames of the displacement. And if you do that, this extracts a nice and robust measure of the global displacement across the entire recording. So that's the bones of our algorithm. And there are a few more bells and whistles that you need to add in order to make this something practical that can be used out in the world. The main thing is to add a non-rigid motion estimation. So the way we do that is by dividing the recording into many soft blocks, running our algorithm within those soft blocks, and then estimating a global displacement, which sort of allows the blocks to talk to each other a little bit when one of them is confused and help each other to come up with a nice non-rigid motion estimate. And there are a few other things that we've added. So for instance, in a lot of recordings, you might see time regions where the activity kind of drops off for a little while. And in those cases, we can use these spatial and temporal smoothing operations to sort of make sure that the motion looks clean there. So when we run this on kind of classic IBL data, you kind of see a variety of motion profiles in that data. So from very stable recordings where you want your motion estimate to be basically 0, to more rarely some recordings which have certain artifacts or non-rigid drift effects. And what we sort of end up seeing is that over the large corpus of IBL data, DREDge kind of makes small improvements over kilosort, which I'm summarizing in this plot here. Maybe I won't go into what exact metric is being plotted there just for time. So these recordings typically have very little drift, so the improvements that we can get are quite small, but they add up over the scale of thousands of recordings. In the imposed motion recordings that they made with the NeuroPixels Ultra probe, we found a sort of different story where kilosort was sort of struggling to model that activity over time. And we sort of think that this might be because this probe is shorter. It turns out that there are fewer neurons being recorded in the smaller area of the probe. And that means that if there's a relatively large amount of drift relative to the length of the probe, you'll have neurons coming in and out of the recording, which makes it hard to model these recordings with one single global template. And that's the kind of case where our algorithm is sort of designed to handle that. We also found some interesting results in chronic data. So here, this is a recording. These are SNPs from chronic recording by Anna Lebedeva from the NeuroPixels 2 paper. So these are 4 shank recordings. This is just showing data from one of the shanks. And what I'm plotting here is between these dashed lines, 10-minute SNPs from recordings across different days. So here is a sort of timeline of these recordings. The scale bar there is 20 days. So these are happening over a period of a few months. And it's amazing. You can see really similar population activity across months in these probes, just looking at these spike localization rasters. So we ran our algorithm on this concatenated chronic data. And what we found is if you use our algorithm's motion estimate to correct motion over days, and then you sort of just go in by hand and grab spikes that live in well-isolated spatial blobs, you can find really well-isolated unit-looking things. So this sort of convinced us that over time, even though the population being recorded is changing, and there's plenty of drift in the individual unit waveforms over time, some of the units are stable enough that you can just grab them directly using this kind of simple criterion. Although really sorting over days is, I think, a more complicated thing that needs a subtler approach. So we also think that because the activity pattern and the population patterns are changing over days, it's another case where having local information rather than trying to use a global approach can lead to improvements, and we tended to observe that. Here's another experiment that we tried. So this is in the NHP data that we heard about this morning. So as part of that project, at some point, I'm not sure why, I'm not sure who decided to do this, but maybe it was Eric or Andrew. They set up the recording to run during the entire probe insertion, going all the way from the brain surface all the way down into basal ganglia. So they're really making a recording over several microns of brain tissue, which is really amazing data. And if you plot a raster of the spike activity over time, it's amazing because you can see individual units sliding across the entire depth of the probe. So every unit that shows up in this recording is there for 5 minutes or so, and it moves across the entire surface of the probe. So it's really cool data. Impossible to model this with a single global template because no neuron is there. But we found that our algorithm could kind of unroll this entire trajectory across the entire insertion. And what you end up getting after you do that, so here on the left, I'm plotting the unregistered, uncorrected spike positions. So this has a scale bar of 500 microns, reflecting the typical dense layout in a neuropixels probe. Here, in the next one, the scale bar has changed dramatically. So here I'm plotting the motion-corrected spike positions over time. So this is kind of unrolling this entire column of neural activity. And it's visualized, it's very compressed and hard to see here, but if you zoom in, you can again see kind of well-isolated blobs that look like single units. And in our paper, we kind of go into looking at what's in those blobs a little bit. But one thing which is cool that you can do is for every spike, we can also grab some kind of features of the waveform that that spike had. So we can grab the peak height and the peak-to-peak duration, other classic electrophysiological features, peak-to-trough ratio, and things like that. And these are things that we know vary and are correlated with the type of the cell that's being recorded. So if you do that and then you plot distributions of those features over this entire column of activity, it sort of can confirm what they thought the probe trajectory was going to be during this really long insertion. So you sort of see boundaries in those feature maps that sort of match when they expected to be transitioning from white matter to striatum and things like that. So this is kind of, we think it could be a cool way for experimentalists to get another channel of information to confirm the trajectory of the probe, or even just to generally map the brain they're inserting the probe into, understand individual variation in brain anatomy, and that kind of thing. And it's also just fun to look at. Okay, so those were some examples of success stories. And then I want to circle back to this human data. So this is really challenging data. Just looking at the spikes alone, it's hard to say that any cross-correlation based algorithm would be able to do much with this activity. It's very noisy. So previous methods that people used, one method I'm thinking of is by Duo Xu and the Chang lab, is a sort of semi-automated program where you go manually into the LFP band and trace out the motion there. And that's what worked really well for them in their spike sorting. And that kind of told us that maybe the motion was present in the LFP band and something that you could track there. So in collaboration with that group and also with a group at Mass General Hospital, especially Angelique Polk, who is an amazing scientist working there and had her own method for tracing motion in the LFP band, we sort of came up with an automated approach to track motion from LFP. So here what I'm showing is a heat map visualization of LFP coming from a human intraoperative recording. And human data is super different from anything I've seen in mouse or other cases. So this is LFP band activity. Here I'm showing depth along the probe, and the x-axis is time. And it has these amazing oscillations, which are very clearly visible. And they make you wonder, if these up and down oscillations, which are happening at a sub-second time scale, are corresponding to actual physical motion of the probe or what they are. And it turns out that they really are, and that the visible motion you can see there is coming from actual heartbeats and breathing. And it's just because in humans, they have to make relatively large craniotomies, and our brains just pulse with our heartbeats so much more than in other organisms. So you get this really huge and rapid motion of the brain surface relative to the probe. So this here, each individual oscillation is, something on the order of a couple of 100 microns, which is, the kind of magnitude of drift that you would hope not to observe at all in a typical acute recording. But it turns out to be salvageable. So, and they already knew that using their manual methods to track this motion. But we can do the same approach that we use for spike raster plots. We can cross-correlate each frame of this LFP activity here at a much higher temporal resolution because we're sampling these frames much more rapidly. I think what I'm showing here, the frames are coming in at 250 hertz in this resampled image. So we can cross-correlate all of those frames, find the best spatial offsets between all of the pairs of frames, and we get this cool oscillating displacement map. And we also get a correlation map between all of these frames. And it turns out that in order to solve the motion estimation problem, the correlation in this data is really important. So if you run this algorithm in a non-correlation aware way, you get the output that I'm showing in pink here, which is very noisy and unstable. But if you make use of this correlation when solving our optimization problem, which tracks the motion, you get a much cleaner output, which really tracks the oscillations that are visible in the data. And we can even do this in a non-rigid way, just like we do in spike data. So if you do that, and if you apply it to the same data that I was showing, that very noisy scatter plot from the beginning of the presentation here, it's really crazy that these really well isolated single units show up. I would never have guessed that there were, maybe 10-ish units here that can be really cleanly isolated. So we were pretty happy to be able to track this motion at a fast time scale. And our collaborators have kind of taken this and ran with it. So if you use the LFP to track the motion at a high temporal resolution, and then you use that motion estimate to then correct spike positions, it really reveals these units which you can grab and study. And this is something they've done. So our Harvard and MGH collaborators have used this LFP-based correction. Then they go back into the AP band. They do the same kind of interpolation that DREDge does to correct the motion in the AP band. And so now they're running the motion correction at like hundreds of frames per second. This lets them spike sort and do lots of cool single unit analysis in humans. And I definitely recommend checking out their papers. It's really groundbreaking work with neuropixels running in human data. You know, they're able to track many units during a speech task and study how individual single units are selective for different kinds of phonemes and all of these really cool things that haven't really been possible until they started recording with neuropixels in human. So it's really cool work that they're doing, and I recommend looking up our collaborators, William Munoz and Angelique and Domikos Mazena, among others. Yeah, they're doing cool stuff. They also looked at what happens if you correct the LFP band itself. So if you apply the same interpolation to correct the motion visible in the LFP band, you know, before doing that, the power spectrum is all spread out. So here I'm looking on the y-axis, I have depth along the probe, and on the x-axis, frequency, and we're looking at the power in different bands as a function of probe depth. And if you motion correct this, it reveals a lot of structure that was just sort of flattened out by the drift. So once they did that, they were able to go, you know, find that, you know, these channels were differentially responding to different stimuli, and I'm sure they're going to do a lot of LFP science with that. I don't think their paper on that has come out, and they haven't told me exactly what they're doing yet, so we'll see. I'm excited to learn about that. Okay. Yeah, so I think it's important to talk about how long it takes to run algorithms, because nobody likes waiting. So for our spike-based method, the runtime of this algorithm is basically entirely controlled by the spike detection and localization steps. So here, this is the runtime of the actual cross-correlation and motion solving part of the pipeline. And basically the entire time is spent detecting peaks and localizing them. So this sort of like cross-correlation part of the algorithm is relatively cheap, and it can be swapped in and out for other similar algorithms for free, but they're all spending a lot of time localizing and detecting peaks. So that's what's slow right now. You would think that the local field potential band method would be slower because it's cross-correlating so many frames. So if you're doing that registration at 100 hertz or something, the number of frames that need to be compared with each other becomes absolutely gigantic. So to kind of control that, you know, it's not actually necessary to correlate every single pair of frames. There's plenty of information locally, and we don't actually correlate all of the pairs of frames in the spike data either. You only need a few. And for LFP data, we can implement a sort of streaming flavored algorithm, which goes chunk by chunk in an efficient way. And this is faster than real time. It takes maybe a quarter real time to estimate motion. I think this was running at 125 frames per second or something like that, which is much higher than the typical motion, but in humans, it's very necessary. So yeah, that's pretty much it for that tool. This is something that's in Spike Interface. At least the Spike-based version of it is easily available in Spike Interface. They have this cool high-level function which lets you basically swap this into kilosort dynamically without, you know, needing to worry. For other cases, if you want to use the LFP-based method or do something low level, there's this GitHub repository, and there's also some MATLAB code that our MGH collaborators have worked up to plug this into kilosort, the LFP-based interpolation. Cool. So next I'll talk about some of our work in progress spike sorting methods. I think we don't have like a concrete spike sorting product that's available yet, so I'm just going to talk about some of the ideas, and we're hoping to come out with a beta version this summer. But for now, I'll just share some ideas. So kind of the driving force behind this is sort of building a drift-aware sorter. So it's pretty common, you know, in real data for there to be drift-induced spike sorting errors. So one flavor of those are, you know, recordings where maybe your motion estimate wasn't correct. And what happens then is that a unit which lived on one set of channels at the beginning of the recording will be interpolated, should be interpolated onto that same set of channels by the end of the recording, but it's not. And that leads to, you know, this unit here, I think, is one of these examples. This is taken from the IBL visualization website, where there's a very similar template, but the channel neighborhood is slightly different. So this unit gets split into two. So we want to correct those kinds of errors with better motion estimation. And we also kind of want to think about waveform changes over time, amplitude changes over time, and come up with a clustering method that can handle those cases. So we're working on this pipeline called DARTsort. It starts with our detection and waveform denoising steps that I briefly mentioned at the beginning of the presentation, followed by motion estimation. Then there's a sort of drift-aware clustering step. And finally, a template matching step, very similar to the one that DREDge uses. So for our detection and denoising, we kind of have an approach that's based on a previous spike sorter from the Paninsky lab. And the idea is to do what we call collision cleaning using a neural net. So this is really similar to the idea that DREDge uses when it's doing template matching. If you have nice template waveforms for multi-channel waveforms for lots of units, you can find the ones that match the best, subtract them away, and that leaves behind other ones which are then easier to see. But the problem is you need the templates first, and you need to sort spikes to get templates. So, you know, can we find a way to take advantage of that idea before we know the template waveforms? And the way we do that is using this neural net to denoise the waveforms. So what we've do, which is probably too small to see, I'm realizing, is we can train a neural network which learns to predict the template waveform from raw data. So it takes in a raw waveform and denoises it, not just removing the, you know, thermal noise in the recording, but also removing colliding nearby in space and time spike waveforms. So once we do that, we can then subtract those denoised waveforms away and reveal the smaller ones, very similar to a classic template matching, but before we have the templates. And this turns out to help a lot of our steps. So for instance, if we're looking at these localization scatter plots, using the neural net by itself already is very helpful in denoising the spikes before they enter the localization operation. That really cleans up the point cloud. So if you apply the neural net, that can reveal these single unit blobs in the localization point cloud much more cleanly than they were without the neural net. And if you also then do our iterative subtraction procedure of getting rid of the largest waveforms before grabbing the smaller ones, that also helps reveal single units a little bit more cleanly. So that's one component of our sorter. And then another main idea that we're working with is kind of an alternative approach to drift handling. So in DREDge, the approach that's used is to interpolate the recording according to the inverse of the motion estimate. So that means kind of averaging the activity between neighboring channels after you displace them according to the drift estimate at each time. And what that means is you're basically taking data which can look like this. So this is some noisy neuropixels data. Sometimes it's cleaner than this, and sometimes it's not. You're going to end up averaging across these different channels, which can be quite noisy. And as we heard from the NeuroPixels Ultra talk this morning, we also know that spikes can be really narrow. So if you imagine a unit that lives, a narrow unit that's being picked up on one of these channels, And if you're sort of averaging it with neighboring channels, you're going to be cutting its amplitude in half and sort of reducing the overall signal to noise ratio of the recording. So we sort of think that this sub-channel spatial interpolation, maybe there could be an alternative approach to drift correction for these recordings. And our approach is just don't interpolate the recording. Instead, let's interpolate our templates. The template, these things are nice and smooth. So when you interpolate them, you don't risk damaging your signal to noise ratio in the same way. And you can do it in nice ways to preserve narrow units. So this is our idea for an alternative approach to drift correction, is to keep the recording as it is and shift the templates around when you're searching for them. And in order to do that, you need to be able to do a clustering of features which are motion corrected so that you can get those templates to shift around in the first place. So we sort of are working on methods for clustering, under sort of shifting channel neighborhoods in a fairly simple way, and then computing templates in a way that handles the drift. And also, of course, on top of that, we have amplitudes and waveform shapes, which are changing over time. So we are kind of building this method for extracting clusters in a drift-aware way. So here is a little movie of some clusters drifting around. So this is a similar plot where I have probe horizontal and vertical positions in these two channels. The left panel is colored by spike amplitude in a rainbow color map. And here we have the cluster identity. And on the right-hand side, the horizontal axis is amplitude of the spike. So we sort of see these clusters moving around in amplitude, and they're being visualized in a motion-corrected way so that although the recording itself has some motion in this case, that motion is being corrected before making this visualization. Yeah, so those are kind of the ideas that we're building our new method around, and I hope we have some more stuff to share on this soon. Yeah, so thanks. This is some links where you guys can find our code. And I just want to thank all of our many collaborators. The DREDge paper has many, many amazing co-authors who shared data and collaborated with us on working on it. So I want to thank all of those people. Thank you, guys. Yeah. Yeah, I mean, so I've looked at some mouse LFP recordings, and they just look qualitatively really different from the human ones that I've seen. And I have no idea why that is. I'm sure that there's lots of amazing things that our human brains are doing to make that LFP look really cool. But I know that it's also really an intricate procedure to do all of the grounding and referencing in the room to make sure that you're getting LFP features. And it's something that human practitioners really care about for clinical reasons. To them, LFP is like the main thing. So they're really careful when they're doing their recordings to get the best LFP they can. And I'm really curious to like explore and maybe learn from people here what the landscape of LFP recordings in non-human data looks like and whether that's possible. To me, it's a bit of an open question. I wonder maybe in non-human primates, there might be some utility there. When you get it in humans, it's so clean and easy to use. So I hope that it's there in other data as well, but I'm not sure. Yeah, I've looked at some IBL mouse recordings, and the method doesn't work as well there. The LFP looks much more diffuse, and it's harder to get a hold of. So yeah, I'm not sure, but I haven't found a use for it outside human yet. But I wonder if there is one or why there isn't. If there isn't, maybe somebody can educate me here today. Yeah. Thanks.
In one slide at the end of the DREDge, you showed how, like, the very complex one was recovering the drift. And
For the human one, this one?
And on the top one, there was like those horizontal lines that you said were maybe.
Yeah.
They don't appear.
Yeah, so they're down here. So these two horizontal lines, they're getting spread out inversely to the motion estimate. So they become fainter, but they're still there. So everybody should use Olivier's tools, which are available in Spike Interface for detecting and eliminating dead channels in their recordings. Not like me, who didn't do it in this one case. Yeah, no, I mean, it's possible that a unit could stick to something. I don't think so in this case, but yeah, thanks for the question.
And I wanted to say, it's not really a question, but there is a bug in the DREDge one, where the LFP and AP traces are not synchronized. There is like kind of a millisecond delay.
Oh.
And there is a way to catch that and make sure that it's synchronized. I mean, I think 1 millisecond from what you're doing is nice.
I'll check, because I wonder if my collaborators who are working on this human data might be aware of that. And I wonder. Yeah. Yeah. That's a good point.
It's a bug that I found that I talked with Jennifer Colonel, but she said that indeed she found the bug also. They still don't know the ones.
Yeah, I think Jennifer Colonel is a clearinghouse for spike sorting issues and bugs, and she's amazing. Yeah. Cool. Maybe not.